Let's start talking about these subunit polysaccharide inactivated vaccines. How does this polysaccharide differ from the protein variant? Well, to answer that question, let's start with a bacteria here. So we're just going to take some bacteria, and maybe this bacteria has a capsule. Okay, maybe this bacteria has a capsule, all right? And let's just say that this is a polysaccharide capsule. When I say polysaccharide, what I'm basically saying is that the capsule around the bacteria is made up of carbohydrates. It's not made up of proteins. Remember, proteins are peptides. Polysaccharides are carbohydrates. Okay, these are different substances, different macromolecules. So what I'm saying here is, if I'm thinking about making a vaccine for this bacteria, do I want to make a vaccine where I give the patient the whole bacteria? Maybe, right? That might be, an option, giving them a live attenuated vaccine, but maybe I don't want to give them the whole bacteria, right? I don't want to worry about this thing potentially infecting my patient. So I just say, look, I'm just going to give them the polysaccharide. Okay, so if I just give this patient part of this polysaccharide capsule, maybe that can generate an immune response to produce some antibodies. Okay, so let's think about this. Can the dendritic cell see this polysaccharide as an antigen and then present it on MHC2? Well, here's the problem, right? A dendritic cell that may recognize this polysaccharide, for example, it's not going to be able to put it on MHC2. And you might be saying, well, why is that? Why can't it just display it there? Because remember, MHC only displays protein antigens. Okay, that's something we talked about. It only displays protein antigens. This is not a protein. This is a polysaccharide. So I cannot display this on MHC2, which means if I cannot display this on MHC2, I cannot activate the T cells. I can't generate a memory T cell response, which means I can't then cause class switching to occur, which then means that I will only have IgM antibodies responding to this polysaccharide capsule. Now, in real life, it's a little bit more complicated than that. But remember, if you have B cells around that recognize this polysaccharide capsule, they can still make IgM antibodies, right? They can still do that. However, you might get an inflammatory response potentially, but it's not going to be a strong one. It's not going to be a super strong memory response, because if there's only polysaccharides around in this capsule, then this is going to be a T helper independent response. And we talked about this. This is only going to generate a humoral IgM response, not the IgG responses that we generated with all of these other vaccines. So this is going to be the weakest immune response that we've seen thus far among our vaccines, just from a general perspective. And the classic example of this is going to be the strep pneumo vaccine PPSV23. And this vaccine is also sometimes called pneumovax. Okay, so this is sometimes called pneumovax. And the reason that it's given, you might be saying, well, why do we even give this thing at all if it's so weak? Well, it covers a lot of different variants and strains of strep pneumo, okay, 23 of them to be specific here. And so that's why pneumovax is sometimes given. There's 23 different serotypes that we can target. But the big thing is you start to get levels that decline after 4 to 5 years, depending on how robust your immune response is. And so you need very frequent boosters. And so that's kind of the catch. Because you're generating this IgM response only, you're going to need even more frequent boosters, even more frequent than you might need for some of these other vaccines. Okay, so that's something to keep in mind. There is a way kind of around this for certain vaccines where what we can do is we can take this polysaccharide and we can say, look, I can maybe attach this polysaccharide to something that might be able to get through this pathway. I might be able to attach this polysaccharide to a protein. Okay, because if I can attach this polysaccharide to a protein, then I can get this whole thing potentially presented to an MHC2 receptor. Okay, because then we have a protein component with it, right? And this thing here is essentially going to be my protein. Okay, so the protein, I can pick any protein that I can bind this polysaccharide to, and this whole thing together is going to be called a conjugate. Okay, so this whole thing, when I combine these two, this is going to be called my conjugate. Okay, so I'm essentially conjugating, I guess you could say, a protein to a polysaccharide. Okay, so that's essentially what we see down here, the polysaccharide plus the protein. And the idea is, now that I have this protein and this polysaccharide here, I can put that on my MHC2, because my MHC2 can certainly display the protein. Okay, and then I have my polysaccharide here. And now these T cells can essentially bind to this MHC2, and we can still generate antibodies with class switching, right? Because now I can go down this pathway and I can get IgG antibodies. So I can get a humoral IgG antibody because I have a protein component now that this dendritic cell can display. And again, will I generate a cellular immune response? The answer is no, right? This thing, which is part of a bacterial wall attached to a protein, dendritic cells will phagocytose it, but the dendritic cells will not be infected buy it per se. And so the classic example of the vaccine here is going to be the PCV13 vaccine. And the PCV13 vaccine, this is sometimes known as the Prevnar vaccine. The easy way to understand these vaccines that fall into the polysaccharide / conjugate category is just remember, if you have a patient and they have a splenectomy, what are the organisms you vaccinate for, right? It's these organisms because they all have capsules, polysaccharide capsules. Okay, so you can see strep pneumo, meningococcal vaccine, right? Haemophilus influenza type B, right? And again, strep pneumo for patients that don't have spleens. And we already kind of talked about that, so I won't go into too much detail on that. The last thing I want to talk about here, just really briefly, just be familiar with, some of the RNA vaccines, specifically the mRNA vaccines. We're essentially taking, a strip of code, putting that in our host. Okay, so let me kind of illustrate this here. So instead of putting in a polysaccharide or a live bacteria or virus, what we're doing is we're putting in a strip of code. So I'm just going to kind of put this out here. So this is just some strip of mRNA. Okay, and so this strip of mRNA is going to have a specific code on it, and we're going to put that in the host. The host will have cells that take up that mRNA, and our cells will then generate specific proteins from that mRNA. Now, in the example of COVID-19, quite simply, we'll generate spike proteins. Okay, so our own cells are making the spike protein from this code of mRNA that was given to the host, right? And our own cells take up that mRNA and we start translating that mRNA and start making these proteins here. So we start to make these spike proteins, and then we make the spike proteins, right? Our own cells make them, and then we make antibodies to the spike proteins that our own cells made. So that, I know it's kind of confusing, but again, this is not super high yield at the time of this recording, although I do think that it will start to become asked about a bit more frequently, as this is certainly a global phenomenon right now. And again, the classic example for this would be the SARS-CoV-2 virus or COVID-19. The last thing I want to say is that for some of these vaccines, especially the polysaccharide vaccines, you can give things like adjuvants. Okay, so you can give things like adjuvants. And an adjuvant, essentially what this is it's something that will allow for there to be a higher, stronger, more robust immune response. And we don't have to go into a ton of the details on this because it's kind of in that medium low yield rating. I just thought I'd bring it up. But the big one to be familiar with when you're talking about adjuvants is aluminum. Okay, so aluminum is the one, if you're going to remember any, you give the patient aluminum inside of that vaccine or as a component of that vaccine, it can tend to boost the immune response under specific situations. So just something to be familiar with. But hopefully now you have a little bit of an understanding of these different vaccines. And I always just go back to the simple pathway. If you're ever in doubt, right, is it a protein? If it's a protein, you know it's going to go on MHC2, right? If it's not a protein, it's not going to go on MHC2. If it doesn't go on MHC2, you're not going to get class switching, right? That's the concept. If it's live and it's infecting cells, you're also going to get a cellular response. Because if this dendritic cell gets infected, CD8 positive T cells will also be activated. So that's the concept. Remember which vaccine goes where. Remember polysaccharide, just think, what are the things you vaccinate against if you have a patient that has a splenectomy? Those are the vaccines we're talking about. And then I think you can pretty much, I just remember the subunit protein here as pretty much Tdap with hepatitis B in there. And then the whole inactivated versus the live attenuated. I think a lot of these make a lot of sense because we don't give live attenuated influenza vaccines. And then I already talked about identifying polio, right? Remember the K in Salk for killed vaccine or whole inactivated versus Saban, right? And so I think some of these will make a lot of sense once you go through this once or twice.